I'm Gert-Jan van Ommen. I used to be head of department of the Leiden University Medical Center, Department of Human Genetics. I have been working on genetics and genomics and the development of therapies and diagnostics for rare diseases and later also common diseases. In a later stage, I have been involved in the Dutch and European biobanking field. Biobanking is the whole field where people collect large amounts of samples to study small effects and try to find out risk factors for frequent diseases. We developed a technique called exon skipping for one of the most frequent rare diseases, Duchenne muscular dystrophy. That's a disease that affects 1 in 4,000 newborn boys. And the boys affected usually end up in a wheelchair in their teens and usually die in their 20s and sometimes in their 30s. And the technique we developed, called exon skipping, actually works by hiding the mutation and in such a way allowing the genetic code to be read on and creating a functional protein still that is partly functional and in this way cause a milder disease. That technique has been developed in competition with a few other people in the world and is still very much in emergence and can be used for other diseases as well. And our department still works at developing this for other diseases like Huntington's disease and CADASIL. The collaboration started as a licensing collaboration, but quite rapidly it grew into a much more intensive collaboration that has gone on for close to 15 years. In fact, it was both. We have been working on Duchenne for the better part of 3 decades. In the late 80s, we were together with our competitors in the hunt for the Duchenne gene. After that, we developed mapping techniques and diagnostic techniques for it. And in the late 90s, we started thinking about developing therapeutic possibilities for the disease. That was when we started with the exon skipping technique, and by 2000, 2001 we had shown that it worked in patient cells, and that was the moment when we decided to file a patent for it. In parallel, at Leiden University, there was a company established by 3 people from the previous Dutch biotech scene, led by Gerard Platenburg, called Prosenza. And their aim was to apply molecular biology techniques for 2 medical problems. I was working at the time with a biotech consultant, Otto Postma, and he knew them from their previous activity in the Dutch company Farming, and he brought us in contact. And they were very enthusiastic about our approach, our field, the chances of success because of the successful pilot experiments. And so that was the point where we decided to team up. Developing a therapy for a severe human disease for which there was no medical intervention available. And so that was one. In addition, we had been funded by the Dutch parent project, the patient and parent organization for Duchenne, from the early days of our work. And they also insisted that we should go towards therapy a bit more, and they were very enthusiastic about this additional opportunity to get the expertise of biotech entrepreneurs. Well, as said, we fit their aims very well. They wanted to do more or less precisely this, and we had the proper and deep expertise on the scientific part, and they expected that their expertise on the translational part would just complement the whole thing. All the time and then some, because the work we did was the major new thing in the Duchenne field itself. So besides helping this development, it was also in the direct line of our own scientific goals. So it was what we did. In talking only a few days and concluding dinner, in practice perhaps a few months for getting the first license agreement signed. On the other hand, there are some challenges afterwards. Small startup companies need to watch their budget and just don't run along too fast. And so in the further agreements, when we developed the work further, they took often their sweet time to translate that into agreements. And that means that you're developing a lot more knowledge and value than you actually have agreed to share, which of course you're nonetheless doing. And so there you are being, in a way, you're being held hostage against your hurry of getting on with the work. And I don't think, when looking back, that that's a good idea. It should be taken much more stepwise, that you develop new things and that you then make the next agreement and so on, because in our case it has worked out fine, ultimately, but it took 6 years after the first agreement to have a real good envelope agreement for everything that we had. And in that time there were sometimes frustrations and negotiations and renegotiations, and I think that this could have done better. There are only advantages because of the complementarity of the activities. Scientists don't make drugs, they develop proofs of concept, and biotech companies then take this to the market. And in this case, we had our scientific expertise, developed the readout technologies, understanding the disease and what to expect. And on the other hand, the company could garner venture capital for much more serious funding that was quite necessary, and actually argue with the regulatory authorities on the permissions for the trials. I will never forget, I think, the historic moment when the first good laboratory practice medicine came in at a price tag of $300,000, and then it turned out that the regulatory authorities first wanted us to order 3 of those batches to just prove that they were all 3 the same. That's a reasonable thing to demand when the development is near the market and you want to prove that it wasn't a single good luck event. But so early in the trials it was impossible, and it was simply too expensive and doable. Very much so. It's now 16 years on and we still work together. We've learned a lot. we brought the field much farther, and we laid the basis for a technology which is still in its infancy, even after, well, like 15, 20 years, but has a lot of promise. I would think so. We've been working together for 16 years, and many of the experiments, most of the experiments went very well. They are applying it now for other techniques as well, so that the basis of our technique really has a lot of promise in the future for several diseases. Not a lot, actually. Perhaps be, as I suggested earlier, a bit more strict in keeping the scientific advances and the research in step with the agreements. That's a good practice, I think. And as an overall advice, especially for people developing therapeutics, I would suggest go and talk with the regulatory authorities as soon as you can, perhaps even in the design, well, preferably in the design stage, because they have very strict demands. It needs to meet requirements that academics often don't foresee very well, and you wouldn't want to know how often applications like this for regulatory approval simply won't work because of ill-designed trials. And if anywhere, then it's notably in this stage where the clock and the meter starts running like crazy, and before you know it, you have a setback of 3 to 5 years because you have to do something different that you could easily have done right the first time.